Thank you, Neboisha. As Neboisha mentioned, I'm going to talk to you about how we've used selects really to try to identify novel guide RNAs and use them to probe sort of guide RNA for CRISPR-Cas-based systems, structure function relationships, as well as hopefully also expand the universe of DNA sequences that can be efficiently targeted for editing. And the work I'm going to describe for you today was really spearheaded by an excellent grad student who's getting ready to finish named Corey Bush, with help from two other graduate students, Jonathan Zhao and Temo Lalanga, and also with some support from two great research scientists, Amos Jan and Juliana Laser. And as Neboisha mentioned, we're all at Duke University. So quick disclosure, Duke has filed patents on sort of the compositions of guide RNAs I'm going to mention, as well as their use, and finally the methods of generating them, and I'm listed as inventor. So when we began this work, really the challenge that we wanted to try to address is that I'm sure many people in the audience know, when we would make CRISPR guide RNAs to say target a gene of interest, we would see, a couple would work out of 10. We'd make 10 and say 2 or 3 would work very efficiently. And that was despite sort of good, using sort of good algorithms to try to predict ones that would work well. And so, that was a bit frustrating, particularly if you wanted to hit a specific part of a gene, say for not just knocking out something, but if you wanted to edit it by, specific base editing or particularly prime editing. And so this inability or the lack of ability to generate guide RNAs to many DNA target sites actually limits potential applications of editing. So when you start to think about this problem, one of the main drivers of this, we believe, and others, is that if you have your guide RNA Cas binding domain, which I call an aptamer because it's essentially an aptamer, the part of RNA that binds Cas9. You basically take that and you're basically swapping in different DNA targeting sequences. So again, here's an example of say 2 different ones. So you swap in target sequence 1 with this, you get a guide RNA that folds up. And if you have this fold into a correct scaffold that lets you assemble sort of this functional complex of, the Cas9 with a correctly folded Cas9 binding domain, stuck to the DNA targeting domain that gives you access and can bind DNA and then cleave it, or depending on how you modify the Cas, it can edit it in different ways. So this is what you hope happens, is that you get correct scaffold folding, but unfortunately, we in the aptamer field often know that as you stick different sequences on the ends of aptamers, you can get incorrect folding of these things because it can influence the proper folding of this, such as shown in this pathway as shown here, that if this target RNA folds back in properly somehow onto this other, the aptamer binding domain, you can get an incorrectly folded scaffold, and then you're not going to be functional. And so we wanted to try to address, could we generate new aptamer domains that would facilitate for different, say, target site 2 here, that would facilitate now getting a functional complex. And so we wanted to get to this question, could there be more than one guide RNA scaffold? So here's the secondary structure for one of the Doudna reviews of the SP-Cas9 guide RNA as shown here. And people have done in the literature a number of rational mutagenesis studies of this, where they would truncate things like they would show you could truncate stem loop 3, shown here in sort of yellow. This would hurt activity but wouldn't kill it. But there hadn't been really sort of real randomization and try to understand what you could change or not. And so we wanted to approach this with our background in selects and with aptamers, saying how much could we really change of this? And this really reminded me, I made my students go back and look at the Turk and Gold 1990 paper, because it was basically a very similar idea as what Larry and Craig did back then, to say, what were the sequences that were involved in looking at the T4 phage RNA protein interaction. And so the idea was, if we could change this, we didn't know if you could change it at all, if it would be very locked in sequence wise or not. Then if you could change it, could you use different guide RNA sequences then and pair them with different DNA targeting sequences and hopefully get things that would work better? So I'm going to show you how we went about trying to attempt this. Probably not to show it for this audience, but just sort of remind everybody the general selects approach that, again, Craig and Larry published again way back. Basically, we wanted to make a library of aptamers or potential aptamer sequences. We had incubated with our target. In this case, it would be Cas9. Select the guys that bound from those that didn't and amplify them as shown here. Now, in this case, we wanted to make our pool, again, based on this guide RNA sequence. So what we decided to do was keep, and as I'm sure most people know, when you make one of these libraries, you need constant regions at both ends. So we decided to keep the 5' in constant region, a specific sequence which targeted a part of the GFP gene that the wild type guide RNA was known to be able to target well and edit efficiently. So we kept this fixed. And as I mentioned, you can actually lop off the stem loop 3. It doesn't kill activity, it actually reduces it some, but we decided to keep this fixed. So again, our 2 fixed regions are shown here sort of in the boxed area. Now there's about 70 some nucleotides between here, as you can see. We recognized that we couldn't totally randomize this and really probe this library efficiently. So what we decided to do instead of making a totally random library was make a degenerate library based on this sequence. And the way we did that is sort of outlined here on the right part of the slide, where again, so here's our fixed sequence called the guide, which is the DNA, spacer, which is called the, it was really the DNA binding domain of the library. But say the first nucleotide, if it was supposed to be a G, shown here, what we did was in the partially randomized library, we made that 58% of the time a G, and then we made it a degenerate library, we put 14% of the other 3 nucleotides there. We did that at each position throughout this strand here to make a degenerate library versus a totally random one. And then what we did with that was we basically did 3 stages of functional selects to try to get things that would work as novel, guide RNA. So we took our degenerate guide RNA library shown here. Initially, we just did standard selection where we incubated with Cas9, formed complexes. We partitioned the complexes from the free RNA by passing it through a nitrocellulose membrane. The complex remained bound. We eluted them. The free RNA went through, and we amplified them, very analogous to what Turk Gold did way back. But we did a few cycles of this selection, I think 2 or 3 initially, and we said, well, we also want to make sure that we're not just forming a complex, but this can bind DNA. So we did a second stage of selection, which is to say, okay, let's throw in DNA. And we knew that once this complex formed, it was pretty stable so that we can biotinylate the DNA. So then we will pull out things that can form a complex on the DNA, the RNA, by then using a streptavidin magnetic bead to pull out these complexes. So you form the complex, you pull it out with a magnet, and then you go back and you isolate the RNA. So we did a cumulative of about, I think it was 2 rounds of this and 3 rounds of this type of selection. We just cloned and sequenced and picked sort of like 200 sequences and tested them for cleavage. And unfortunately, only about, I think it was less than 5 showed much cleavage activity. Even though the pool, you could start to see a little cleavage activity. And so we knew that there were active new guide RNAs in there, and there was still a lot of variability at this point, but very few of them were functional, even though they were probably binding. So we decided it was probably important to add a third stage of selection, which was more of a functional stage. And actually the student, Corey, came up with this great idea. He said that when this complex forms, it's known to be stable. And actually that it exposes a free end, a 3' end of this cleavage complex. And so he rationalized that he could use terminal transferase to say, add a poly A tail to this. And so this would mean that, that you'd actually gotten cleavage there. And then he would use an oligo DT primer to pull out actually the cleaved DNA, the complexes that can cleave DNA. And so then he could pull these out and then isolate RNA and do that. So what he ended up doing was 2 rounds of selection like this, 1 of this, and then 2 rounds of selection like that. And then what he would do was just follow to say, was this enriching for cleavage on these rounds as shown here? And you could simply do this by a DNA cleavage assay. You could also do it, I'm not showing it, actually by looking just by bead binding. You could show you were enriching the ability to pull out more DNA if you fluorescently label the DNA over these cycles too. So here on this gel, it's just basically it controls the first 2 lanes. Here's the uncleaved DNA. It's basically the DNA with the guide RNA without Cas. You throw Cas9 in there and you get nice cleavage like everybody knows, these 2 products. Round 0, which he doesn't show here, there's basically no cleavage with the pool. Round 1, or it's just doing binding the protein, you may see a hint of, you know, some cleavage when you incubate that. Round 2 and 3 again, where you're also doing with DNA binding, again, you start to see some cleavage, but it's, you know, it's still minority. And again, you're throwing in a vast excess, actually, the guide RNA here. But round 4 and 5, you can see by doing this functional selection and the TDT tailing and pulling out, you can see sees a big increase in the ability of these pools now to cleave DNA. So we were excited about that, went on and then did high throughput sequencing of those cycles. He got 300,000 approximate sequences to begin with, and then started to do informatics to say, how could we group them and analyze them by similar sequences or frequency, and thinking of things, the secondary structure, et cetera. By doing that, I'm going to show you how he tried to bend these into families. There were approximately 650 different phylogenetic branches of a tree. And interestingly, I found out right away, I didn't know if we'd mainly get the wild type guide back, because it was by far the most abundant thing to begin with. And we knew it was functional against the sequence, but it wasn't even the most abundant guide that came out. It was the 4th most abundant of the sequences he got, and only represented 1 or 2% of the total number of sequences. So that was interesting to us. But what was increasingly or even more interesting, if you start to look at the sequences that come out of the selection. So on the top of this slide, here's the native or the wild type guide scaffold sequence shown here. And then he's just aligning, this is about half of those 650. It's just too much to put on one slide of those groups. What he's showing here in a color code is the change. So if it's black, it's identical to the native or the wild type guide. But if it's changed, it's, say, for example, these A's are changed to C's in the blue and all of these family members. And what you would see is what was nice from like an aptamer selection, again, you would see this phylogenetic covariation. So you would see these all go with C's, and again, these guys would all go as G's, implying or again demonstrating, just like you see in nice aptamer selections or like what we see in evolution of ribosomal RNA, again, you just need a base pair here. And so if you start to walk down through all these sequences, it's interesting because you can see you tolerate a lot of changes. So most of these guys had up to 20 nucleotide changes of the original, you know, approximately 70 or so that were randomized. And the closer you got to 3' and you saw many more changes. So if you start to look at that as far as identity compared to the prototype or the native wild type sequence, here's the 58%, you know, level that we put in the original library. So you see some of them actually prefer another nucleotide besides the wild type, that the wild type isn't preferred, as shown in these guys. And then there's a fair number that, you know, you don't really care. They're close to this 58%. And then there's a bunch more that say, well, you don't really have it 100% conserved, so you can tolerate some things at these positions. There's very few that actually require, or it's really at least difficult to change from the natural wild type sequence. So we took really a subset of these to start to characterize in more detail. So we took 130 of them. Some of them are sort of indicated by stars. So we try to take sort of ones that were part of these different phylogenetic branches as representative ones, start to characterize them more. So we tested 130 of them initially just for cleavage in vitro. And it turned out most of them, approximately 100, cleaved comparable to the wild type, at least with the scaffold, at least with the targeting the DNA sequence and GFP we did the selection with. And so if you look at those 100 here, the subset, again, this is a similar type of analysis, and you say, how similar are they to the wild type and how much can you change them? Again, you see there's a lot. Here's this 58% line where your pool started. Again, there's some that don't like to be the wild type sequence at all. And there's a lot that you can actually change. And there are very few, again, that are up here around 100% that you can't change very easily in the guide RNA sequence. So that was interesting to us, and we wanted to say, well, where were these guys that you couldn't change? So we mapped that onto the crystal structure of the guide RNA bound to Cas9. And so this is actually the front view, so-called front view of the guide RNA within the context of the CRISPR, the Cas9 structure. Here's the back. But just to orient you here, so shown in purple is actually the DNA strand that you're bound. The black here is the 5' end of our library. It's also the DNA binding domain, again, of the guide RNA. And then you follow that around here, and here's the guide RNA, the part we randomized, you can see, and here's the 3' loop that I said you could kind of lop off, and we kept it fixed. Now, what's interesting, we've color-coded here the guides in red that are really hard to change. And what you can see both here in the front view and in the back view, they all cluster right around where the cleavage is happening of the DNA, sort of near the active site, essentially, for at least the nuclease in the protein. Maybe that's not surprising, but again, it was really neat to us to see that there were really only a few, less than or about a handful, that were really required specific sequences. And then the rest, as you march away from there, you can see there's a few that are, you can change a little bit, like 90 to 95% like to be the wild type. And then you march away. Yellow, you can change a fair amount of the time. And the green, actually, you don't care very much. And so from these results, again, it really suggested that there are very few nucleotides in the guide RNA that you can't change, but you probably need a secondary structure. Like I was saying, you need base pairs often through here, but you don't care what the base pairs are to position these handful of nucleotides in the right position to facilitate cleavage. So from a structure-function relationship, we thought that was incredibly interesting, but we wanted to go on and say, okay, of these 100 guys that we know were active, how many of them worked in cells? And so since these were all targeting GFP to begin with, we did a simple, obvious experiment, which is to say, let's make RNPs of them, transfect them into cells, and ask how well they edit, or in this case, it would be knock out GFP, make indels compared to the wild type guide. And again, I remember I told you we picked a sequence in GFP we knew the wild type worked well in. So you could see it can knock out, in the best cases, like in HeLa cells, over 90% of GFP by a simple transfection like this within 2 or 6 days, depending on if it's a short-lived GFP experiment or a long-lived one. And we did this in 3 different cell lines as shown here. And what you saw was that some of the novel guides, and these again have up to 15 nucleotides changes compared to the wild type, were essentially as good as the wild type. You couldn't get much better because this guy was targeting very well to begin with. And then you saw some of them didn't work as well at all. So again, there were some that came out of the selection that were equivalent, and other ones that didn't work as well, as shown here. And if you try to depict all of this, so this is looking at these 130 different novel guides now, shown phylogenetically here. And here's the wild type shown here at 3 o'clock on the thing. So it has 0 changes from the wild type. But as you walk around here, again, this phylogenetic tree, on the inner circle, the color coding is saying how well these guys work for in vitro cleavage. And again, the bright red says they basically work as well as the wild type guide. So you can see most of them work as well as the wild type guide. And some of them have, again, 15, 16 different nucleotide changes from the wild type. We didn't test them all, but you can see we tested about 100 or almost 90 to 100 that for in vivo or intracellular cleavage. You can see there was a range of activity. They didn't necessarily always work. If they worked in vitro, at least against this one target sequence in vivo, we don't totally understand that, but many, about a third of them, also showed good editing and knockout efficiency inside of cells. And again, what was interesting about this is some of them, again, had, say, this guy, 96, worked as well as the wild type, and it has 16 nucleotide changes from the wild type. So there was a number of them that diverse sequences that could affect, similar editing as in vivo, at least in cells, as the wild type. So that was interesting, but we knew it would be tough to improve upon targeting that particular sequence. But we're going to get back to this initial issue, which is, okay, now we had a bunch of variable novel aptamer scaffolds for guides. Could you use them to enhance targeting to sites that the wild type guide didn't target well? And so we just scanned through the GFP gene because we had this all set up. We chose 5 additional target sites. Turned out the wild type edited them from 40 to 95%. This is sort of similar to what you see a range of editing when you just randomly pick sites in a gene. The 95 was the one we knew worked well that we started with, which is shown here in red. So that was sort of our, and that's target number 1 is shown here. Again, the wild type works well against that. Again, here's a number of these. We tried, I showed some of this data previously, but again, some of them work similar to the wild type, and then some of them don't work well against it at all. And it's hard to do better than this, but here's a second target. So this guy the wild type scaffold only edits 40% of the time. And you can see, hey, we swap in different scaffolds that have different aptamer sequences. And now you can get ones that are over 80%, approaching 90%, whereas the wild type can't do that. And again, interestingly, some you see are much more effective against a particular target, like the scaffold 205 is almost as good as the wild type against target 1, but doesn't work well against target 2. We think that's probably because it folds poorly with the target 2 DNA binding domain and folds reasonably well with the target 1. So we went and did this in more detail. So we picked 5 target sites as I was showing here, and 10 different of the 30 some that we knew worked well guide RNAs, and we basically, this is a heat map of this type of study, and we looked in 2 different cell lines to do this in more detail. So target 10 was actually, it's the 10th potential site that's targetable by the SP-Cas on GFP. Again, we knew it works well. So again, the wild type worked against it pretty well, and there were a couple, as you can see here, that were essentially nearly or basically as good as the wild type at targeting that site. So red here is very high cleavage efficiency, sort of black is not particularly good, 50%, and then white is it doesn't work at all, like negative, or we didn't try it. So if you go to like a second target, so target 5, again, the wild type doesn't work well against it, not particularly. And again, there were 3 different novel guides that edited it much more efficiently, getting up near 90%. It was interesting for this target 12, nothing worked very well. We didn't, at least of these 10 we scanned, couldn't get anything that worked well for it. Target 19, actually the wild type was the best. We didn't find of these 10 any better. But then in the last 2, target 21, this guy worked better. And then target 32, this guy worked better. So in really 3 of the 5 new targets, the novel guide scaffolds were more effective and significantly more effective than the wild type sequence. So we think that means you can use these novel scaffolds now to hit additional sites that are hard to hit, which is the wild type sequence. The other thing that was really interesting to us is we saw that certain guides were much better at targeting a particular DNA sequence than others. So just for example, if you zone in on this example of guide 226 versus 232, they basically totally swapped what they were able to hit well. And so we wanted to look back at this secondary structure map, this tertiary structure map, where those changes were in the guide RNA. So to reorient you again, here where again the DNA strand is for cleavage is in purple. We've made the whole guide RNA now in red, but we've color coded the variations that you have in the 226 mutant in yellow and the one to the 232 mutant in blue, and then there's a couple where they both change. I'm showing here in green. And what immediately is apparent is they're changing totally different regions of the guide RNA, the wild type guide RNA. And again, and they work better with different targeting strands. And we think that's again totally consistent with, depending on what sequence you put here, you want to get rid of negative interactions that may occur in different parts of the guide RNA. So we thought that was really interesting, that you would change different parts depending on what target sequence you wanted to put up there. So in summary, what we're very excited about is that we figured out that the select strategy, again, from the 1990 paper, really can be used to generate novel functional guide scaffold variants. And we've already identified over 100. We didn't sample all of them. Again, that was 130, but we have tens of thousands of them. That it turns out from this analysis, even initially, that very few nucleotides, a handful, are highly conserved. You can change most everything else. That different guide RNA aptamer domains can pair more effectively with different DNA targeting domains to create scaffolds with varying degrees of activity, and that selective scaffolds can enhance editing at DNA sites that look like that are challenging with the wild type native guide RNA sequence for SP-Cas9 at least, and that we think that generating novel guide scaffolds and methods to create them should be make more of the genome tractable to efficient editing. And I should point out, we didn't, for those alternative targets, do selection with them as the 5' fixed region. And so we think if we go back now and switch that constant region and select against that, we'll even be more efficient, be able to get out variants that will work particularly well against them. Because obviously the way we did it, we're addicted to the initial 5' targeting sequence for the DNA. And so we think the approach should be very amenable and let you generate now editing machinery that can target many, many more sites. And then finally, just acknowledge the people, and I did it at the beginning, and thank you.